biographies! Yes, plural this week, because when you start talking about something as multitudinously awesome and elegant as DNA, you have to wonder just who figured all this stuff out and how big was their brain? Well, unsurprisingly, it actually took a lot of different brains in a lot of different countries and nearly 100 years of thinking to do it. The names you usually hear when someone asks who discovered DNA are James Watson and Francis Crick, but that's bunk. They did not discover DNA, nor did they discover that DNA contained genetic information. DNA itself was discovered in 1869 by a Swiss biologist named Friedrich Miescher. His deal was studying white blood cells, and he got those white blood cells in the most horrible way you could possibly imagine, from collecting used bandages from a nearby hospital. God. For science, he did it! He bathed the cells in warm alcohol to remove the lipids, and then he set enzymes loose on them to digest the proteins. And what was left after all of that was this snotty gray stuff that he knew must be some new kind of biological substance. He called it nuclein, what was later to become known as nucleic acid. So Miescher didn't know what its role was or what it looked like. One of the scientists who helped figure that out was Rosalind Franklin, a young biophysicist in London nearly 100 years later. Using a technique called X-ray diffraction, Franklin may have been the first to confirm the helical structure of DNA. She also figured out that the sugar-phosphate backbone existed on the outside of the structure. So why is Rosalind Franklin not exactly a household name? Well, two reasons. One, unlike Watson and Crick, Franklin was happy to share data with her rivals. It was Franklin who informed Watson and Crick that an earlier theory of a triple helix structure was not possible. And in doing so, she indicated that DNA may indeed be a double helix. Later, her images confirming the helical structure of DNA were shown to Watson without her knowledge. Her work was eventually published in Nature, but not until after two papers by Watson and Crick had already appeared, in which the duo only hinted at her contribution. Even worse than that, the Nobel Prize Committee couldn't even consider her for the prize that they awarded in 1962 because of how dead she was. The really tragic thing is that it's totally possible that her scientific work may have led to her early death of ovarian cancer at the age of 37. At the time, the X-ray diffraction technology that she was using to photograph DNA required dangerous amounts of radiation exposure, and Franklin rarely took precautions to protect herself. Nobel Prizes cannot be awarded posthumously. Many believe that she would have shared Watson and Crick's medal if she'd been alive to receive it.